The overall goal of this time-lapse recording is to observe the intracellular transport events in Caenorhabditis elegans neurons.
This method can help answer key questions in the cell biology field, such as axonal transport and intraflagellar transport. The main advantage of this technique is that one can observe intracellular transport in C. elegans without dissection.
Start this experiment by growing transgenic C. elegans strain of interest at 20 degrees Celsius on lawns of E. coli OP50 feeder nematode growth medium plates. To prepare agarose pad for worm mounting, first mix 0.4 grams of agarose in 4 milliliters of distilled water in a glass tube. To avoid evaporation, close the tube with a lid. Then, put the tube on a 95 degrees Celsius heat block for 90 minutes. Fill one more glass tube of the same size with distilled water. Keep the tube at 95 degrees Celsius and place a pasture pipette in it. Next, use this warmed pasture pipette to place 2 drops of 10% agarose on a glass slide. Before the agarose drops solidify, quickly cover them with a second slide while pushing the slide to flatten the agarose. Let it stand for 1 minute. To avoid clogging of the pipette, quickly flush it by pipetting 95 degrees distilled water up and down until the agarose is washed out. Then, leave the pipette in 95 degrees Celsius water. After 1 minute, the agarose pad between the glass slides solidifies. Carefully slide the slides apart by hand.